pop them into chat just like normal. I will send the PowerPoint also in a little bit across for all of you. We've got a lot of stuff going on today to cover. We've got your generic things like your units and everything, and then on top and standard form. And then we're going to go on to looking at all of the different tests that we do, such as like Simpson's diversity index, the chi-squared test and all of that stuff. And we're going to be doing a little bit of logarithms and some of the biological exponential graphs in that kind of area as well. So if you've got anything else that wants to be covered that I haven't mentioned, come to the top of your head, put a little message in the chat, and we'll also go through that today because I think we'll have time. Okey-doke. So these are your conversions that you need to know. They're really, really simple. Sometimes people just forget to know them in the last minute. The ones that you need to know are your kilo, your deci, your centi, your milli, your micro, and your nano. So pretty much everything on that list other than the pico at the end there, I believe. Please don't forget them. Please don't forget how to change in between them because they're such easy marks to be able to get. They can come by as a question by itself, or it can be like, can you convert? You might have to convert the units at the end of a question. So it's a really easy 1 mark to pick up or 2 marks to pick up. So if you can make sure that you know all of these different numbers, know the symbol and know the prefix, that'd be really good. Okey-doke. So these are our common biological units. You're expected to understand what they mean. You're expected to know in what situations they're coming up to. I will send you the PowerPoint, so don't worry if you don't need to like screenshot or anything. So we've obviously got your distance and your area and your volume. Everyone typically knows. Just know that milliliters is equivalent to centimeters cubed, and microliters is acute to millimeters is equivalent to millimeters cubed. That's just an important one. Density is always mg per centimeters cubed. That is really useful to know because it helps you remember the equation for density as well. So you can, like m and g, you can figure out what it is, and then obviously divide it by the centimeters cubed. It just, from the units, it helps you work out what the equation could be. For instance, if you have forgotten, with the microbiology, you've got a colony forming units and our plague forming units. They're normally done like colony forming units per minute. So you normally get a little graph that says something like that. That's just what that means. Heart rates, everyone should hopefully know. Okey-doke. So with standard form, obviously everyone knows that it's a mathematical notation. There are 2 rules to standard form. Obviously, you can see I've got A times 10 to the n. So A cannot be bigger than 10. So A must be sort of like a single digit number. And obviously n also is meant to be an integer as well. Okay, so you should all have a calculator of some kind. This is just how to convert from decimals into standard form, or vice versa. Most people have the Casio, so it's the S to D button on your calculator. If you can't find it, it's really easy to do a quick Google. It will let you know all on there. Okay, measuring quantities by difference. Again, you have your temperature difference given to 1 decimal place, so you have to match the resolution of the measured values. The 0 here would be a significant number because you're trying to match up those decimal places. So it's just really important to make sure that all of your significant figures in your decimal places are matching up. Percentage change, we've got a little question here, so feel free to have like a minute or so to answer it. Pop your answer into the chat if you'd like to as well. Yep, so well done to those people who put the right answer into the chat. It was 12.5. We have our equation here. You're expected to remember this equation. It's not given to you. I've got a huge list at the end of the PowerPoint of all the different equations that you need to know, so you can bear that in mind going into your next exam if you don't know already. Well done to everyone that answered. Okay, percentage yield. Have another go at this question for me. I've got the equation up there for you. Don't get that in the exam, but I've just forgot to get rid of it. So have a little go here for me. Yeah, well done to everyone who said 75. There we go, it's at the top there. Okay, phenotypic ratios is something else that you come across. Some people find this easy, some people find it hard. If you cannot remember these straight off the top of your head, for example, like a certain cross will be 9 to 3 to 3 to 1, always just draw it out and count it up. You literally cannot go wrong with drawing it out, counting it up. It will make your life so much easier in the exam. It's definitely worth taking the time for these questions to just draw it out quickly. It's definitely the smart thing to do. So have a little go at this question for me here. If you don't know, I'll give you a second. I mean, people are already on it, but I'll give you a second if you don't know it off the top of your head. I know some people have memorized these things. I personally didn't for my exam, but just have a go. Okay, yeah, well done for everyone who got that phenotype right. It was 3 to 1, and extra well done for the person who did the genotype as well. So you are going to have to know to be able to do these ratios in both monohybrid and dihybrid crosses. You aren't going to usually be asked genotype ratios. It's normally phenotypic ratios, but just bear it in mind. Obviously, it's going to work the exact same. We're just looking at the alleles instead of the overall outcome of what they're going to look like. Okay, reporting ratios here, it's very similar, but have another go. This is just us taking actual numbers and applying it to maths. Okay, yeah, well done to everyone who has said 3.9 to 1. The thing to remember, and what might catch people out, and I think it might have caught someone out today, is that when it says of red eyes to brown eyes, the second sort of category that they're asking about, in this case, brown-eyed flies, needs to be down to 1. So if they ask brown-eyed flies to red-eyed flies, then you bring red-eyed flies down to 1, and the answer would be, yes, so someone just asked if the answer had to be x to 1, and yes, it does have to be x to 1. I accidentally changed the slide. Yes, I think someone might have written 2 point something to 1, or like that would have been if it was like the other way around, but always the answer is always x to 1. Please remember that, because that's an easy mark to get. Okay, so microbiology dilution. So this is where we can figure out the number of bacteria in the original colony. It's sort of working back, it's sort of going step by step and figuring out the maths. So we're going to go in depth with this. Let me know if you've got any questions along the way. So we're going to imagine we've got a 10 milliliter starter culture of bacteria. It's been incubating, we're doing a serial dilution, and we've got all the figures up there. Then a 10 microliter sample of this dilution. So remember that it's a microliter sample. We're going to then take and we're going to put it into an agar plate, use some agar jelly, and we're going to figure out the individual colonies. So the resulting plate looks like this. So can you first of all tell me how many colonies have now formed on the resulting plate? Pop that into the chat for me. Not the original number, the current number on the plate. Yes, well done, 19. Okay, so in this kind of question, that is the very first step. So that's step 1 of tackling this question. You get a mark for, if they haven't already given it to you, you will get a mark for simply saying there is 19 colonies formed on the plate. something like that. Pop it in there, really, really easy to at least get 1 mark on this question. Okay, the next step is where we kind of get the maths involved, and to work out the original number of bacteria in the sample to begin with, you need to multiply that by the dilution factor used. So if you could have a go doing that right now, I'll put it up step by step at the end for anyone who struggles with this. But if you do know how to tackle this question, I'll give you a few seconds to have a go. Okie doke, so we've got some answers coming in. So what we need to realize, so the very first step here is that we have 19 colonies on our plate. The step then is going to be to realize that we have 19 and 10 in a 10 milliliter, in a 10 micrometer sample. We're going to work backwards from the information that's been given to us. We then need to find out how much from the microliter sample would go into the milliliter culture. That's why we're multiplying by 1,000 here to begin with. And that's where we're getting our 19,000 from. So that's our first step. So now we worked out how much is in the 10 milliliter diluted culture. The next step is to then multiply it by 1,000 because we're going to the starter culture. And I think someone explained that quite well. Because because you're doing 10 to the power of 3. Don't get tricked out too much by the numbers, but remember it's a 2-step thing, so you have to find out what's in the 10 micrometer sample and then work out what's in the 10 milliliter sample. That's why we're doing the timesing by 1,000 and then timesing by 1,000 again, which is where we're getting the 19 million overall. Just remember that that is a 2-step process there. Okay. So applying that same thing, I've got another question here. Have a little go with that question. There is 3 steps to this rather than the 2 we had last time. So it's a little bit harder, but remember, there are 3 things that we're doing in this solution. So there should be 3 mathematical steps, if that makes sense. Okay, I know people got a bit confused from the last one. So this one, I've got more of a picture here, so I'm hoping this might help you imagine the steps a little bit better. So we have 1 centimeters cubed in our original solution, and we're taking out a 30th of that. So then, because we've got like 30 centimeters cubed and we're taking out 10 centimeters cubed, so we're taking out a 30th. And it's then being diluted by a factor of 10. And then we've got another 1 centimeter cube being taken out, and that's being diluted again by a factor of 10. And then we've got 1/100th of the solution of solution 3 being taken out, going onto the agar plate. Does that make sense? That little bit going there. Oh, okay. So if we go back up to the question, we have a number of yeast cells in 13 centimeters cubed of the solution. And then we're looking at the first dilution and we're taking out 1 centimeters cubed. That means of the original solution, we're taking out a 30th of it. So 1 out of 30. We're then mixing it with 9 centimeters cubed. That means that we're having a dilution factor of 10. So like 1 out of the total 10 is going to be in the original. So for solution 2, 1 out of the total 10 is going to be from the original solution, if that makes sense. We're then doing another step of this into solution 3, which again is going to be 1. We're taking 1 centimeters cubed out of solution 2 and we're adding that into 10 centimeters cubed of solution. But because there was already a 1/10 of a dilution in solution 2, by the time we get to solution 3, we're actually like 1/100th of a dilution because we're taking it out and then taking out a mixed solution again. If that makes sense. So the answer will be 6 times 10 to the power of 6. Because we're doing the 100th of the solution multiplied by the 10, multiplied by another 10. So the 100th of this, 100 are coming out from the end in the agar plate, a 10 from solution 3, a 10 from solution 2. Then we're going to multiply that by the original solution, which was 30. And then we're going to multiply that by the 20 colonies grown. I'll repeat that again. We have 20 colonies grown. So we're going to multiply that 20 colonies by 100 because we have 100th of that last solution. We're then going to multiply it by another 10 because we have a 10th from solution 3, and then another 10 because of the 10 in solution 2, and then we're going to multiply that by 30 because of the 30 in the original solution. How do you get to 20? Oh, the 20 is simply coming from the 20 colonies that have grown on the agar plate. No worries. Okay. If you can, in the exam, draw out what's being diluted each time, it will make your life a little bit easier. But all the numbers will be on, I know this is a bit of a pain to say, but all the numbers will be on the page for you. It's just about knowing what you have to multiply together. So I hope that makes sense. Can you go back to last? Yeah, I'll quickly go back to last question because I think a couple people have struggled. There you go. I'll give you guys a minute again for this question. For this question, you have to make sure that you're looking at the units because you have microliters and milliliters, so the units have to be the same no matter what you're working with. And you have to remember that it's a 1 times 10 to the minus 3 dilution, which is essentially giving us the fact that it's a 1 in 1,000th dilution, if that makes sense. So I'll quickly pop over to this question where we had this fact diluted by a factor of 10, diluted by a factor of 10. We're looking at dilution by a factor of 1,000, which is why we're multiplying by 1,000 in this equation. Does that make sense? So the 1 times 10 to the minus 3 is showing us that it's 1,000th dilution, which is why we're multiplying by 1,000. Okay, going back to this question, someone asked another question. Are we timesing 100th 1/1000 of 30 centimeters cubed by 20 centimeters? Okay, where we know that a 100th of the solution 3 is going to be on the agar plate, we can then figure out, so we're doing the 20 colonies on the agar plate, we're timesing that by 100 because 100th of solution 3 has gone onto the agar plate. And then we're timesing that by 10 because a tenth of solution 2 has gone into solution 3. And then we're timesing that by another 10 because a tenth of the original solution has gone into solution 2. And then we're timesing that by 30 because there is 1 cent, because a 30th of the original solution of like the yeast cells has gone into the original solution. Are there any more questions before we move on? Because I know that this is like a very tricky thing to get your head around. So if no one's got it, I will be happy to like explain it again. You can even direct message me. Don't worry about sending it to everyone. So how many marks these are usually? You can get, it could be a good 5 marks depending. So for this step where you have the 20 times 100 times 10 times 10 times 30, because there are essentially 5 different steps that you're doing, you get a mark for each step. But in the actual exam, you might get a couple of like steps given for you, which is where we then can get reduced marks. So on average, I'd say they're normally about 3 marks, but they can go up to 5. Could you explain what it is exactly with 20? Oh, 20 is, so what we're working out in this question is the number of E. coli from the original 30 centimeters solution. So we have to work out that there is 20, that there have been 20 colonies that have grown in the agar plate in order to then go backwards and see how much would be in the original solution. So that's where we're getting the 20 from. I hope that makes sense. I also haven't seen it recently on a past paper, but it's in the specification. Okay. I haven't got any more questions in, so I'm going to move on. We're going to look at logs. Oh, I forgot to get rid of the question. We're going to look at, ignore the at what hour is there, but just what is the number of bacteria at 6 hours? Have a go at this question for me. Please don't make the mistake of not looking at what's on the axes. Okay, don't freak yourself out over this question if you haven't seen it before. All you need to know is just how to use logs. I'm sure you all know that you need to use logs in biology. So I just read off the graph, so I made this question up. So I just went up with the log of the colony forming units is 6.5 at 6 hours. So we're going to go up to 6 hours and we're going to go across. And you need to remember that log of the bacteria equals 6.5. So knowing that information, what would the total, for the grey one, sorry, I didn't specify, what would the total number of bacteria be? So have another go in case you got that one wrong. Okay, yeah, the answer is the 3162277.66. Very well done for those people who got it right. So we've here got that the log of the number of bacteria is 6.5. So how we get the number from that is we go 10 to the power of 6.5. So I forgot to actually type it out on the screen, so I'll pop it in the chat. If log of bacteria equals x, then bacteria equals 10 to the power of x. Yeah, I don't know how, I mean, I know how it works, so I took A-level maths, but you don't need to understand how it works. You don't need to understand how it works or why it works. You just need to know that to get from log of a number equals this, then to go backwards, you need to do bacteria is 10 to the power of that. I hope that makes sense for everyone. If you've got any questions, speak now before I move on. Okay, this, I'm going to, you guys will have it on the chat on how would you find the log from the bacteria total? Oh, hang on, let me go back quickly. Yes, it does work the same as chemistry. How would you find the log from the bacteria? Like, could you be? Okay, you won't be asked to work backwards because this is biology. You're not expected to know that, but you might be given something in the sense of, you know how we had our answer as we wrote 10 to the power of 6.5 and we got our final answer. You might be given the final answer as 10 to the power of something, which means that the of something would be like log of that number. Does that make sense? You won't be given a number and then figure out that's 10 to the power of what, because that's a very maths question. So you wouldn't, yeah, okay. I hope that makes sense. But yeah. Okay. This is, I'll pop it up. I'm not going to go through this. You will have the slide afterwards. You don't have to know the formula. You just need to know the stuff in, like, you can figure it out yourself. I'm sure you'll be fine. Are you covering paper 2 math skills? Yeah, I'm covering all math skills, including paper 2, obviously, because paper 3 as well. So we've got the diameter of a neutrophil being measured, and we want to calculate the volume. So have a little go at this for me. Okay, so I've got the answer up there for you. So the volume of a sphere is 4/3 pi r cubed. R is the radius. So you've been given the diameter is 12 micrometers, so the radius is going to be 6 micrometers. And then from there, you just plug it into your calculator and round it to 3 significant figures. Okay, so next I'm going to be looking at is mean. I'm sure this has been like drilled into us for a long time, so I'm sure you understand how we use it. Oh, okay, someone wants me to repeat that. The volume here, because a neutrophil is a sphere, we have to look at the formula, which is 4/3 pi r cubed. So what r stands for is the radius. We've been given the diameter, so the radius is half of the diameter, so the radius is going to be 6 in this case. So we're just going to plug that into the equation and stick it into the calculator and just write out whatever number that gives us. And then we're going to give it to 3 significant figures. Just in general, whatever comes out of your calculator, no matter how many decimal points, write down that answer before you do any rounding at all. You can even be rounding it to like 4 decimal places just to save you time, but just write out everything so you can ensure that you'll get at least working marks through what you're doing. Okay, so the mean, I'm sure you guys know how to do it. You just add it up and you divide it by the number of things that it's talking about. If you want to find out how to do it on your calculator, because I didn't know how to show you guys myself, because I didn't think you could like see through the screen if I held up my calculator on the camera. So I found a little YouTube video that shows you guys how to do it using the KCO Scientific, like the class whiz calculators, which I think is what most people have. I think if you have a graphical calculator, chances are you to also take maths, so you should hopefully know how to do it. But yes, so in this equation here, we've got x bar. X bar is the x with the little line on top, and that means the mean. The little symbol of E with the x is showing that it's the sum of x, and we're putting that all over n, which is the total here. Just to note, if you're finding an anomaly in your data, you do not include it in the mean. So you have to get rid of any anomalies before you're taking the mean of anything. So in this question, we're just gonna move on to median and mode quickly. Can you just find the median and mode? I'll give you guys a minute because I do want to get on to the statistical tests, because I think that's why most of you are here. Okay, the mode is something what we call bimodal because we have both the 25 and the 31 repeating twice. So the mode is both the number 25 and 31. For median, you need to reorder all of the things into, you need to reorder all of the numbers into like ascending order. So you'll go 19, 24, 25, 25, 27, 28, 31, 31. Then you have to pick the middle value. Now, because there are an even number of values, the middle value is actually going to be the 4.5 value. So it's going to be the value in between the 4th value and the 5th value. And how we figure that out is we actually average out the 2 values that we have either side. So we're picking that middle value either side of the 4.5, like of the 4th and the 5th. We take the 4th value and the 5th value and we work out the number that's in the middle. I hope that makes sense. Okay, correlation. This is what's listed in the specification for OCR, which is very, very similar to AQA, so chances are you're also going to need to know all of these as well. So just have a little look. Okay, magnification is size of image over size of real object. This is one of those, that's one of those questions that you just need to know how to do. One of those formulas that you need to be able to recall. The most important thing is to make sure that the units are matching up, because that's the biggest way that they trick you out with this question, is when they don't have the units matching up. So please remember to make sure that all the units match up. Okay, moving on to statistical tests. We have the chi-squared test. In the chi-squared test, there's always going to be an observed and expected table of results, which you can see up in front of you. You've got the expected frequency and the observed frequency here. When you're doing any kind of chi-squared test, the question you need to answer is there a significant difference between the expected and the observed frequency? So what we're looking at here is the difference, and we need to be able to top that up and compare that to any critical value that's been given to us. What the critical value is telling us is if a difference is comparatively larger or the difference is comparatively smaller than the critical value. If we have a number larger than the critical value, the difference is significant. If not, that's insignificant. For any of you who take maths, I know the criteria is different based on the question, but in biology, always remember that if it's larger than the critical value, the difference is significant. How to calculate the number is always just by looking at that table that they give you and looking at the number that corresponds to 5%. Okay, I hope that makes sense. I haven't got any questions coming in. This is our equation as well. So have a little go at this question for me. Okay, I'll just repeat the stuff about critical values quickly. The critical number is essentially telling us that if the difference is comparatively larger, the difference is significant. I hope that makes sense. And we always find out the number that corresponds. Yes, we're given that. Yes, you're given the equation to 5%. So have a write down of the value if you've solved it. Okay. Plugging in the values we have to do for each, so it's the sum of each of these different things. So we work them out all out individually. You can draw another, what I would highly recommend you do is on the table in the exam, draw in another heading that gives like the frequency of observed takeaway frequency of expected squared all over frequency of expected, so that you can pick up marks for just being able to do sort of each step required. And then you were plugging it all in and we're getting 34.71, sorry. And then the value of the 34.71 is what we're going to compare to the critical value, telling us if it's significant or not. Because there are 5 species given, the dandelion, the buttercup, the daisy, the tulip, and the crocus, what is the number of freedom that we have to look at when we're trying to work out the critical value? If we've been given 5, yes, n-1, so 4. So I haven't got the table up here, so just so you know, the critical value given is 9.488. So because there are 5 categories, we look at the 4th degree of freedom, and we're going to look at the 5%. So you're going to have a table of values, and it's going to be number of degrees of freedom by percentage, and we're going to go across the middle, and we're going to find the value of 9.488. Now, the number we have of 34.71 is greater than 9.488. So what's that telling us about the difference? Is it significant or insignificant? Yes, it's significant because why? Yeah, our value is greater than the critical value. Perfect. Does that make sense to everyone? Okay, fab. This is your kind of conclusion sentence at the end. Right. Moving on to the student's t-test or unpaired t-test. Student's t-test and unpaired t-test are the exact same thing. And then we have a paired t-test. So if you ever see these things, student's t-test is the exact same thing as unpaired t-test. A student's t-test is not the same as a paired t-test. Okay. In an unpaired t-test, the data is not paired, measurements are taken from the same individuals. For example, we're going to take the height of 10 individuals of the exact same shrub, where there is a lot of sunlight in an area where there's little sunlight. So we're taking 2 different measurements from 2 different groups, if that makes sense. So it's the same shrub, but we're looking at different, this is going to be shrub A and this is shrub B. The species is the same, but there are 2 different individuals where this is being calculated. Whereas in comparison, a paired t-test is you're looking at the effect of 2 things from the same individual. So a paired t-test would be taking a human and looking, we've got here, the effect of caffeine on the heart rate of a Daphnia, and then the effect of, like, because that is the same thing, and we're using the individual Daphnia in that situation, it's 2 measurements taken from the same thing. whereas in comparison to the unpaired t-test, there are two different individuals with two different factors being taken. I hope that makes sense. Okay, we're gonna have a go at an exam question for the students' t-test quickly. But before that, I'm going to quickly go over your hypotheses. The null hypothesis is going to tell us that the means are equal. The alternative hypothesis means that they are different. You can write null hypothesis, alternative hypothesis. You can write H0 and H1. It doesn't matter. A two-tailed test is what we're going to use for the alternative hypothesis. You don't need to worry about that too much, but just know that the words two-tailed test and alternative hypothesis go together. So a student's t-test takes a lot because you are going to have to work out like the standard deviation, do a lot of division, take a lot of the means. A lot of the time in the question, they'll break it down into parts, and for part A, they'll ask you to find the mean. Part B will be find the standard deviation, and then part C would be like write a student's t-test for it. So I'm going to give you the numbers here, and using these numbers, could you work out the answer to the question for me? No, for AQA, you don't need to know how to use the equation. This is for OCR. But you're going to need to know the ones that come up later, so sit tight. Okay, yeah, so the answer overall should have been t equals 5.66. I've got a degree of freedom table here because we are looking at two different categories. Instead of it being n-1, we actually have to take the two n's and minus it by 2 because we're taking the total of two different categories, so we have to minus it by 2 at the end. So in this case, the degrees of freedom is going to be 18. And the critical value is 2.1. Because our value of t is greater than 2.1, there's significant evidence to reject the null hypothesis, and we take the alternative hypothesis. So for students' t-test, if the value we get is bigger than the critical value, we reject the null hypothesis. Okay. Paired t-test example is very similar. It's simply just popping in again. As long as you know the equation that we have here, so the D with a line on top is the mean of the difference. So obviously you're going to take the total at the end there and add it all up together. Sorry, I didn't mean to scare you a few days before your exam. We're just trying to cater to as many students as possible. I'll just put that up for a few seconds for any OCR people here. Okay. Standard deviation, I've got up another YouTube video so you can learn how to just do it in your calculator without actually having to do the maths by hand if you're unsure. But have a go at working out the standard deviation here for me. Okay, so our overall answer should have been 45.0. Sorry, you wouldn't have had enough time if you're doing that by hand. That was not enough time, but hopefully you had enough time if you're just sticking values into your calculator. If you have time to come back and check at the end, I'd really encourage you to write out the square root of 12147.43 over 6. I really strongly encourage you to come back and be able to do that. Because you can get extra marks in case something goes wrong, just for doing that conversion into the 40.5, just showing that you've done that step into the next one. So if you have time after checking and you're only using your calculator, please come back to that question and just add in that extra little bit. The X with the line on top is the mean. So X bar means mean. Anything that has any letter has a line on top is meaning the mean. Okay. In Spearman's rank correlation, when we're ranking things, we're always going to have 1 as the highest figure. If there are 2 of the same figures occupying 2 ranks, for example, rank 3 and rank 4, they're both given the rank 3.5. I hope that makes sense. So, for example, in female mean mass, the mean female mass, our first rank is going to be the largest number of kilograms. But because we have 2 values that have taken 40, instead of writing one as 3 and the other one as 4, we're going to write them both as 3.5. If we're asking to find out the rank correlation, you are given the equation, so don't worry. But we're looking at the difference between the 2 ranks and then squaring the difference. I'd always encourage you to add extra lines onto the table. And then we're plugging it into this equation. I hope the equation makes sense, but I'll explain to you anyway. So the RS, which is just the rank correlation, is 1 take away 6 times the sum of the differences squared. And we're dividing that by n, which is just the total number of species given here. I've done the maths for you. So the answer that we're getting is 0.8661, but our critical value is 0.7857. So could you pop into the chat what that means with the evidence, if we can reject the null hypothesis, or do we accept the null hypothesis if our value is greater than the critical value? Yep, amazing, well done. We are rejecting the null hypothesis. And what can we say with 95% confidence? Because we've taken the critical value at the 95% level. Does anyone know? Just that extra little bit you can add on. Yeah, well done. So we can say that with the 95% confidence, there is a positive correlation between the gestation period and the mass of the female, because we're rejecting the null hypothesis. So the null hypothesis is saying that there's no significant difference. The alternative, yes, yeah, exactly, well done. Okay, percentage uncertainty, I think everyone will know. Just remember that if you're having a temperature change where you're including 2 different temperatures in the where you're including 2 different temperatures in the difference in your value, you need to double the uncertainty, if that makes sense. If which is why we're doing 2 times the absolute uncertainty at the bottom there. Don't forget that if you're including 2 values, you have to times the uncertainty by 2. Okay, Simpson's index of diversity. I actually don't know if you need to know this for AQA or not, but this, again, it's just a simple formula. It's, again, just a case of being able to plug it in, and you are given everything to you here. We've got the big N is the total number of organism, and the little n is the number of a particular species. Oh, there you go, it's an AQA. I hope that makes sense to everyone. Amazing. Okay, these are your formulas to know. I'm pretty sure that I have gotten this off of the this is off of the OCR spec. But if you want to know what to know from your AQA spec, literally type it into Google and it will bring up the spec and scroll right all the way down to the bottom of maths, of like type in into your like maths equations you need to know for AQA A-level biology, scroll right down to the bottom and it will tell you what formulas you need to know and what ones you need to be given to you. Oh, let me upload the PowerPoint. Okay, I've uploaded the PowerPoint on the chat for all of you guys. It will be on the WhatsApp later. But just so all of you guys have this now. Are there any questions before I let you all go? At all? No worries. Feel free, I'm around for the next few minutes if you have any questions. Okay, I'll have a look at the AQA ones for you right now if you want to know. Yeah, so I put that link in the chat. If you scroll right down to the bottom, go right down to the end page, I think that they have a list of all the formulas to know. Or it might be, if they don't, I'm sure like physics and maths tutor have something up there. Okay, thank you all for coming. Please keep on asking if you've got any questions. I'll be here for a little bit.